Hi guys, today we're going to talk about neurons, neuromuscular junctions. So this is probably something, really the one thing in this unit that we're going to talk about that you're probably somewhat familiar with. So the cell that you see in this picture, this is a neuron. Okay, so the title of this slide is How an Electrical Signal Passes Through a Neuron. And so these little branch-like structures here, these are called dendrites. And they are the receiving end of a neuron. So they will receive impulses from other neurons. And then the signal gets passed to the main part of the cell where the nucleus and a lot of the organelles are, and that's called the cell body, okay? And then the message gets sent down this tail-looking structure, which is called the axon. And at the very end of the axon branches and has these little knobs at the end, which are called axon terminals. Okay, terminal is the end. So an axon terminal is the end of the axon. Normally the axon is going to have the myelin sheath around it, at least for the neurons that we look at. So you'll see that on the next few slides. All right, let's talk about the myelin sheath first. Okay, so a sheath is like a case that you put your sword into. It's like a casing. Myelin is the substance that this sheath is made of. So the myelin sheath is a collection of Schwann cells. Okay, that's what each of these marshmallow-looking things is called. They're called a Schwann cell. And when you say, is that the same Schwann that said that all animals were made of cells? Yes, it actually is the same guy. And that myelin sheath surrounds the axon of some neurons. When you hear about white matter and gray matter in your brain and in your spinal cord, white matter consists of neurons with myelinated axons. Gray matter consists of neurons with unmyelinated axons. Now, what the myelin sheath does, and the reason that it's there, is because it allows for saltatory conduction. Okay, salta, I was just reviewing my son's Spanish with him the other night and his AR verbs that he had to know. And saltar means to jump. So saltatory has to do with this idea that when a myelin sheath is present, when the axon is myelinated, it allows the impulse to basically jump from node to node to node, these nodes of Ranvier. It allows this impulse to jump from node to node to node to node, and allows the impulse to go down the axon faster. So when you have a neuron that is supposed to have a myelinated axon, and maybe that myelin sheath has been broken down a little bit, like what happens in multiple sclerosis or MS, that's kind of what brings about the symptoms of multiple sclerosis, the sensory issues and the motor issues and the issues with muscles, is because in multiple sclerosis, the immune system is attacking the body's own myelin sheath. Okay, where neurons meet up. So the axon terminal of one neuron will meet up with the dendrite of another neuron. So the axon of this blue neuron is going to meet up with a dendrite of the next neuron. So a synapse is the name for the general area where two neurons or a neuron and a muscle cell meet up, okay? Like what you see here. This whole area is called a synapse, okay? And then the synaptic cleft is the part shown in purple or pink here. It is the space between those two cells, okay? I think I feel like we went over that. We went over that in cell-to-cell communication because we were talking about synaptic signaling. So you've heard of that term synaptic cleft before. A cleft is like a gap or a space, and a synaptic cleft is the space between either two neurons or between a neuron and a muscle cell. Okay? And then the neuromuscular junction is where the axon terminal, so this gold-looking structure or orange-looking structure, that is the end of a neuron, and all these little knobby-looking things are axon terminals. And so in this picture, we are zooming in on this blue box here, and we are seeing the specific area where an axon terminal meets up with a muscle cell. And so that's kind of what we're going to draw and go over on the next slide. If you need to pause my video to run and grab some colored pencils or colored pens, I think that would be really, really helpful here. Because this is an important picture and we're not just going over anatomy or how something looks, we're going over how it works also. So it says, let's draw a neuromuscular junction. Now, I only have a limited amount of space on this slide, but you have an entire sheet of paper. So, you know, you can spread yours out a little bit. There may be some times where I have to erase something and you're just gonna have to, because I wanna write something else, you can squeeze it in. I'm just letting you know, I'm a little short on room here. I'm also trying out a new pen to write with. So if it looks a little weird, it's because I'm trying not to use my finger to draw. I'm trying to use this pen so that I get a little bit of a better picture. So hopefully I won't delete anything by accident. All right. Okay, so this thing that I just drew, let's see if this is any easier. It's not really easier. Might be a little easier to draw tiny things. This is called the axon terminal. So it is the end of an axon. And then this is going to be a muscle cell. Okay. No. Sorry, I'm still getting used to this pen. So the name for this entire thing, okay, it could be called one of two things. The general name for it, because it's where either a neuron and a neuron meet up, or in this case, it's a neuron and a muscle cell meet up. The general name for it is a synapse, okay? The specific type of synapse that it is a neuromuscular junction. So synapse would be the more general name. The more specific name would be a neuromuscular junction. Of course, I'm just abbreviating that. You have it written out right here. What else? Let's switch colors again. Come on. Okay, and then we have this space between the axon terminal and the muscle cell, and that space is called the synaptic cleft. That's the space between these two cells. All right, next. Inside this axon terminal, I'm going to outline this a little bit in gray. All right, I've got to erase this over here. You keep it. It's not that it's wrong. I just need a little bit more room. So I want to clear off some of this. Okay. Back to that. All right. These circles that you see here, those are called synaptic vesicles. Okay, so you know that vesicles are used for transport. These are specialized vesicles that are going to be found in the axon terminal of a neuron. And then within those synaptic vesicles, we're going to draw some dots here in green. You use whatever color you want to use. I'm just trying to be consistent here. Okay, and those green dots, okay, there's two names for it. The general name would be that it's a neurotransmitter. So let me explain to you what that means a little bit. These cells, remember that we said paracrine signaling is when the cells are near each other, but they're not actually touching. Okay, this is a specific type of paracrine signaling that we're doing today called synaptic signaling. We're just going into more detail. So when a synapse is involved, okay, these synaptic vesicles usually contain a neurotransmitter that will eventually be released from the presynaptic neuron into this space, and it will bind to receptors on the postsynaptic cell, whether that's a muscle cell or a neuron. We rely on these neurotransmitters to essentially get the message or the impulse from this cell to stimulate something to happen on this cell. So these are signaling molecules. Now the specific one that's involved here, since we are dealing with a neuromuscular junction, is called acetylcholine. And oops, sometimes that can be abbreviated ACH. However, you can't abbreviate it from the get-go. I need to know that you know this word. Okay, so those synaptic vesicles contain the neurotransmitter acetylcholine. That is specifically what they are. Acetylcholine is a type of neurotransmitter. So I'm not saying those are interchangeable. Chances are I want you to label those green dots as acetylcholine. I'm just telling you that acetylcholine is a neurotransmitter. Okay, so let's talk about what happens at this neuromuscular junction. Let's go back a few slides. So remember that I told you that an impulse is going to come down the dendrites, it's going to go through the cell body, and it's going to go down that axon, whether or not there's a myelin sheath there, and then it's going to arrive at the axon terminal. So that is an electrical impulse. So if we go back to our neuromuscular junction picture, we're going to show that impulse arriving at the axon terminal. Okay. Next, let me erase a little bit here. That's still acetylcholine, that's not changing. You can draw this wherever you want to, as long as it's in this neuron. I'm going to switch to pink here, maybe. Okay. So in the membrane of this axon terminal is a voltage gated calcium channel. All right, so this impulse, remember, is a change in electricity. So when this impulse arrives, it actually changes something called the membrane potential on this axon terminal membrane. For right now, just think of it as a change in electricity. So when that impulse arrives, it is going to trigger voltage-gated calcium channels to open. And there's going to be more calcium outside the neuron than there is inside. So that calcium, once the impulse arrives at the axon terminal, that calcium is going to want to come inside. And so calcium is going to enter that axon terminal, and that calcium entering actually changes the membrane potential on the neuron because you're letting a bunch of positive charges in. So it kind of changes things electrically on the inside of the cell. And that is going to cause these synaptic vesicles to fuse with the axon terminal membrane. And then they are going to release that acetylcholine into the synaptic cleft. Okay, so again, just to repeat what's going on as far as physiology goes, the impulse, this electrical signal, arrives at the axon terminal. That will trigger voltage-gated calcium channels to open. Voltage gating just meaning that we have to have a change in electricity in order for them to open. Once those voltage-gated calcium channels open, calcium will come in. Calcium makes the inside of the cell more positive compared to the outside. That change in electricity will cause these synaptic vesicles containing acetylcholine to move to the cell membrane of the axon terminal, fuse with it, and release their acetylcholine into that synaptic cleft. So remember that that process is called exocytosis. Now, on the muscle cell, you are going to have some acetylcholine receptors. Okay, so the acetylcholine just got released into that synaptic cleft, and acetylcholine is going to bind to its receptors, and attached to those receptors is another type of channel protein. Okay, so let's label a few things here just so we don't get it lost in the shuffle. These things that we drew right here, these are acetylcholine receptors. So I've already spelled out acetylcholine. You know I can spell it, so I just abbreviated it there. And then the blue things that we just drew attached to them, those are ligand-gated sodium channels. And remember that sodium symbol is Na+. So let's say we have some sodium out here, which we will. Sodium is going to be in higher concentration outside the cell compared to inside. So when acetylcholine, these green dots, when acetylcholine binds to their receptors, that is going to open up these ligand-gated sodium channels. And when those channels open, that's going to allow sodium to rush into the cell. Okay? So sodium rushing into the cell, again, kind of like what happened up here with the calcium. You are letting a bunch of positive charges inside the membrane, inside the cell, and it's going to upset the electrical balance just because it was kind of what we call the membrane potential was so negative to begin with, and now we're letting positive charges in, that causes kind of a little disturbance in the membrane potential. But what that's going to lead to, that's going to lead to a muscle contraction. So this is sort of how a nerve can signal a muscle to contract. So the sodium comes in, that's going to cause a bunch of other things to happen, but eventually what we're going to have is a muscle contraction from that. So what I need you to think about is the what ifs. That's what your neurotoxin computer lab is going to be about. It's going to be about different neurotoxins and their effect on the neuromuscular junction. So for example, there's one toxin that you're going to learn about today, with your neurotoxin lab that is going to block, oops, hold on. it is going to block sodium channels. And really, you have two possible outcomes here. Either with the kind of things you're going to see from the neurotoxins, they're either going to cause paralysis because no muscle contraction can occur, or they're going to cause too much muscle contractions, basically muscle spasms and seizures, and like having your, all your muscles in your body contract at once, which isn't good either. So we want to figure out which one of those would happen here. If we have a neurotoxin that blocks sodium channels, that means that even if acetylcholine binds, sodium can't get into the cell. If sodium can't get into the cell, that means you're not going to have any muscle contraction. So that's going to cause paralysis. Okay. If I blocked a calcium channel, okay, that's going to cause paralysis, because if calcium doesn't get in, then these synaptic vesicles are not fusing with the axon terminal membrane, and they're not releasing their acetylcholine. If for some reason acetylcholine can't get released, you're not going to have muscle contraction, you're going to have paralysis. But there could be some cases where the opposite happens. Okay, so instead of blocking this, there's some toxin out there that causes too much acetylcholine to be released into the synaptic cleft. That's going to cause too many of these channels to open and stay open for too long. And so that's going to allow a lot of sodium to come in, more than usual. And in that case, you're going to get something like muscle spasms and seizures. You're going to get, that toxin is going to cause massive muscle contractions in your body. Now, we're concerned about both of them. Both of them could kill you. The paralysis, remember, it can paralyze the muscles that you use to breathe, and so that can kill you. The massive muscle contractions and spasms can exhaust your supply of ATP and possibly make it so you can't have more muscle contractions. So I posted a McGraw-Hill video. I think it's on YouTube, so if you can't watch it on your computer, watch it on your phone. I posted that in Google Classroom so that you can see an animation of this. So you'll want to watch that before you continue on and do your neurotoxin computer activity. Okay? Well, thank you so much. I think that does it for today. All right, see you soon.